Hi, welcome to Research Quest Live. Hello, my friends and fellow scientists. My name is Tracy Collins, and I'm an educator at the Natural History Museum of Utah. And I'm here with Dr. Mitchell Power. So welcome, Mitch.
Hi, everybody. Good to see you, Tracy.
Okay, before we get started, I just wanted to add a couple things. If you're on our Research Quest Live website and you would like to ask a question, you can go ahead and press the Ask a Question button. So don't hesitate, really, we can take questions today. Also, if you're joining us on Facebook, you can ask your questions in the comment section, and we're going to get to as many questions as we can today. All right, so we should probably just jump in. Today in class, I know that we've been working on the last investigation, which is all about the future of the forest. And students have been making predictions, and they've been thinking about some of the models that they were looking at, like the annual average temperature over time and sort of the future of where annual average precipitation will be. So anyways, I know that you were saying that you had a photo that you were kind of thinking about sharing with us, and maybe that will help us get started.
Sure. Yeah, so the hardest thing when we move into modeling and the future is that, predicting is a dangerous business, right? Because predictions can be wrong. But they do give us some guidelines of how systems might behave. So, I always like to remind students, take those models with a grain of salt, but use them to form ideas, right? One of the things that has come up in that fourth investigation is the fate of the lodgepole pine trees, right? And so we, been looking at lodgepole pine and what exactly does lodgepole pine need to survive? And we know what's kind of neat about lodgepole pine is that it's like a weed species. It's adapted to poor nutrient soils. It can grow in like sandy, disturbed areas. It doesn't need a lot of the fertility that a lot of other tree species do. So the fact that it has some of these higher tolerances that some weed species have, it actually gives it a little bit of a leg up on what might happen to it. So maybe I'll share a slide here, Tracy, if that's okay. Let's see here if I can do this. Share screen. I'll show you a couple maps today. So can you guys see this map here?
Yes, I can. We can see
Okay,
that.
so this is some model output that was done by a colleague of mine, Pat Bartline. And so these are Latin names for different tree species. And you're looking at, in the center of each map is Yellowstone National Park. So you can kind of orientate. So Utah's down to the south. You can see a lot of Montana and Idaho to the north and west. But so in these different maps, it's showing climate models looking 100 years forward or towards the end of this century and trying to determine, which trees will be more successful and which ones will potentially contract their range. So, we call this the winners and the losers, right? So lodgepole pine is that first one in the top left. It's Pinus contorta. Maybe you can see my mouse moving here. And so, what's really interesting about lodgepole pine, the way we read these maps is blue is new and red is dead. So if you see lots of red, that's bad for a species. If you see lots of blue, that means that species could really expand its range. So if we look at lodgepole pine, green is effectively no big change. So around Yellowstone, not a lot of change. If you look in, you can barely see Utah and the Uinta Mountains in the bottom there. Mostly no change. You might see some contraction towards the lower elevational limits. Now, what's kind of neat, I'll just stop sharing for a sec there, is that each tree species will have a different response. So it's not like, we can assume everything is going to be either bad or good. You have to really look at each species, whether it's a tree or a tree squirrel or a pine beetle, and each organism has tolerances. And as our students have learned, these are what we call, some of the abiotic and biotic factors, right? There's biological factors that influence species success, and then there's non-biological factors. And so the climate modeling, like when you use a model, many times models, especially when they're looking at climate, are just the abiotic, so non-biological features. So it gives us an estimate of what might happen, but think about seed dispersal, or other species like Douglas fir or aspen that might compete with lodgepole pine. It doesn't give us a lot of the dynamic of competition and biotic relationships. It gives us a lot of abiotic. So kind of a long answer. Prediction is a dangerous game to get into about the future, but we can use models and we can look at the past and what we know about how a species behave in the past and kind of project those forward. And under future climate change, we know temperatures will increase. Moisture is really hard. Like myself, I'm not a modeler. I work with modelers, and modelers consistently are challenged by modeling moisture. So where's the rainfall going to come in larger amounts? Where's rainfall going to create droughts? And for any folks out there that have friends or family in California, I mean, you guys have seen droughts firsthand. And the latest news is that California may be moving into one of these 20-year droughts, and certain plants are really going to be challenged by that. So, so I guess if I could just sum all that up, is take each species or each organism kind of at face value and look at the organism and what are the things that control each organism, and try to think about how changes in the future might affect that organism. That's kind of the best way to get a handle on who might be the winner and the losers as we move forward. So did I answer some of your questions, Tracy?
Yes, I think so. Yeah, no, that was great. I mean, I think that one of the things that, like you just said, it's a dangerous game. It's very challenging to make predictions. And one of the things that students really focus on are patterns, right? This idea of patterns and how there's some reliability within patterns. But of course, I would think, as with the weather, right. It's like gold star if you can predict the exact weather 5 days in a row, right? I mean, we know, right. Like we have some ideas, and they're based on these predictable patterns, like you just.
And that's a really good point. Like this idea of weather and climate, right? They're two very different concepts, but the weather is what you and I experience every day, and it's how we relate to each other. It's often the first thing we get to talk about in a conversation is like, hey, it's so nice out, or it's terrible out. But the climate, which is what we like to study, these are the long-term patterns, like you're saying. These are patterns in weather. And so it's really important in your mind to try to separate weather, which are really short-term kind of variations in our climate, versus the climate, which is this longer-term average condition. So, of course, climate's changing, so our long-term average condition may not apply in a few years from now, right? Because we're in a transitional climate period. But yeah, so weather, short-term, it's the day-to-day, week-to-week, month-to-month. But climate, like when I take a climate study and say, okay, here's the climate, I'm usually talking about 30 years of data that kind of summarize the climate of an area. So 30 years of information is all packed in, and so all the weather changes are all packed into that climate. So it's climate is an entirely different beast than weather.
Yeah, no, and that's a great clarification. And I think, in the data that we were looking at today, you look at these annual averages, and it's just so interesting, right off the bat, to see the great fluctuations from year to year, but that we could see these patterns, these big picture patterns over 100 years. One of the things you just touched on is actually sort of my next question, is that ecosystems are always changing.
Yes, they are.
In the video, you really pointed out it's the rate of the change. And so thinking about, the question is, how can we tell when change is normal or if it's something we need to worry about? And like, how do we know when it's time to intervene?
Right. And so, some of you have heard this word a lot lately, unprecedented, right? So we like to think about this in when we study organisms and climate, like which parts of change are unprecedented. And the way we can start to do this is going back in time. So we can use archives of data. We can use weather stations that have been around for hundreds of years. Of course, in the longer weather stations are those from Europe. As you come to North America, we have 100 years of weather stations. So we take all that information, we go back in time, and we try to match it to patterns we're seeing with organisms and movements and dispersal. And so, the rate of change, this is like we like to think of it as the elephant in the room, right? So if things happen so fast that, imagine a tree on a landscape, and that tree needs to make some seeds, send those seeds out, have some babies, let those babies survive and become a tree themselves. Well, the most vulnerable time for organisms is in the juvenile stage. So adults, they gain more protection. Like imagine a big old tree versus a tiny little seedling. That big old tree can handle a really dry summer. The seedling cannot. The seedling will die. In fact, there's some evidence out there that for like Pinyon pine and some of the pines out west, 10 or 12 days of dry soil and they die. So that means if you want to increase populations, you need to take care of the babies, right? The seedlings and the young. And when change is happening really quickly, one problem is can dispersal and reproduction. So can you get those seedlings out or get those juveniles out and secure their population, or is the change happening too quickly for them to survive? So there's those kinds of issues. We can also look back at fossil records. If you go all the way back to the ice ages. So when I talk ice ages, we're 20,000 years ago. And so 20,000 years ago, there was a large continental ice sheet over most of Canada. So these are big changes. And so when that ice sheet melted, we can watch how species recolonized Canada. They basically were following the melting ice sheet back into Canada. And so we can use that rate to see how quickly species move across the landscape and recolonize. And you can get to some interesting ideas like, spruce tree can move 200 meters per year with all the right conditions. And so you can start to put numbers to the rate of movement. And that's going from like an ice age to a warm period we have today. And that change happened over several thousand years. And so now we're having a similar change, except we're going from a moderately warm period to a increasingly warmer period, and that's being compressed into just a few decades. So plants and animals don't have 1,000 years. And the added complexity of this rate is barriers to dispersal. So if you're a pika, or you're a migratory animal and you decide where you live, it's just getting too warm, you need to be able to have a corridor, right? You need a pathway to get out of your environment so you can get into a more comfortable home. And when you add highways and cities and urban projects, we create barriers. So the rate is concerning a lot of scientists. And there's even some fun ideas out there that maybe we should be like trucking organisms around. And since they can't get there fast enough, maybe we put certain species in trucks and move them there, transplant them, let them establish, because we know from some of the climate models that the future climate will be better in certain spaces. So we could put them there ahead of time and try to skip the problem of the vulnerability of juveniles and seedlings to rapid change. So it's an interesting and really challenging area of science to think about rate of change, and then how do you mitigate? Like, what solutions can we have? Yeah, good questions.
That answer you provided really ties in nicely, actually, with my next question about interventions, because a lot of people, I think, wonder, some interventions that have been done in the past maybe didn't play out the way that people thought they were going to play out. And so I'm wondering, what are your feelings about interventions? And then also you mentioned the seedlings of lodgepole pines. Are people currently doing anything to try to save those lodgepole pines? So that's kind of a two-part question.
Yeah, okay. So intervention, it's a very active field, and it's not one that I do a lot of my own research in. And there's mixed feelings, I think, in the scientific community. And so rather than focusing on individual species and trying to protect species, much like we do with the Endangered Species Act, and we try to see, populations are very small, let's protect certain species. Rather than focusing there, what I'm seeing with intervention is focusing on ecosystem function. So, when I say function and we talk about lodgepole pine, for example, the function of lodgepole pine is that it's a tree that can colonize poor soils. It comes in often after a fire. It provides habitat for a whole host of species, for nesting birds and the grazers that live in the understory. It's a habitat, right? And so if you think about function, it's like, okay, if we lose a species, is there another species that has the same function? And so we may actually be better off, since we are losing species, to think about how do we protect function of ecosystems. And if they're functionally doing the same thing, like cleaning our air, they photosynthesize, they produce oxygen, they provide habitat. If they're doing all those functions, yet we're starting to lose species, maybe that the kind of band-aid we put on it is save the function and potentially lose some species in certain areas. Now, I'm not advocating for losing any species, of course, but I think a lot of the conservation efforts are try to think about function, and can we keep function of systems, and whether it's a tropical rainforest or a lodgepole pine forest in Utah, how do you keep the function? So, and
yeah,
then
that sounds.
Your second part of that was.
You had mentioned planting lodgepole pine seedlings.
Right.
If that's, I guess, an intervention that people are currently working on, or if there's something else being done to save lodgepole trees.
I haven't seen or heard much about lodgepole pine, and I think a bit is because it's so prolific. It does quite a good job on itself, planting, and with the increase in wildfires that we've seen in the last couple of decades, there's been lots of opportunity for lodgepole pine recruitment. So we have more lodgepole pine success. There is, with other species, I've heard some discussions like the western larch tree, which we don't have much in Utah, but it's really common in western Montana. It's an amazing tree because it's like the pine trees, we call it a conifer or gymnosperm, but it loses its needles, which is unusual. But the western larch tree, there's talk about taking that tree and moving it around the west into areas where the future will be a better growing condition for it. So there are some ideas about, maybe moving certain trees that could potentially have a range contraction, so they diminish their population numbers. Let's push those into areas where they could then expand their range. And there's too great of a distance to get them there naturally. So those kinds of interventions I've heard about. I think, lodgepole pine, I called it a weed species. It's a tough tree, and I think it's going to be okay. I don't have a lot of concern for lodgepole pine, at least at this point. Yeah.
So great. Well, so we have about 10 minutes left, and I'm really enjoying this conversation. I think we could jump into a little bit more of your research and work.
Sure.
And I have a question here about what are some of the scientific processes that you use to understand the past?
Yeah. So, the approach that I like to take is, we try to find the best evidence to answer the questions that we have. So where do we get evidence? And my interest is, how do forests and vegetation communities change over time? So we have to get evidence, right? And so the evidence for changes in forest are plant fossils. So we have to uncover plant fossils, and then we start to build a timeline. So we use some radiocarbon dating techniques, so we can date old plant samples through the amount of carbon decay in their cells. And so we create a timeline. We look for these fossils. A lot of my work, we study pollen. So we're actually studying microscopic plant fossils of the pollen plants produce. And we then kind of create, it's like a story. Sometimes I tell people it's like CSI, because we have no idea going in what we might find. But once we get the samples under the microscope and we can start to build a story through time, we can see some big changes. So one example I'll give you, which is the lodgepole pine in the Uinta Mountains. So we went out and we did some studies in the high elevation of the Uintas, where lodgepole pine grows today, to determine when did lodgepole pine show up. Has it always been in the Uintas? We know there was big glaciers there 20,000 years ago, so the glaciers must have pushed lodgepole pine out. When did it show up? So we created the timeline through some radiocarbon dating. We collected some of these long sequences of sediment records, and we sampled for pollen. Well, it turns out lodgepole pine showed up only about 6,000 years ago. So if you think about that, lodgepole pine can live maybe 100 to 200 years. So, now you're getting down to, 50, 60 generation times. So 6,000 years is not very long. I mean, you think about longer lived species like bristlecone pine, we've talked about in other Q&As, and bristlecone pine, the oldest pine in the West, that can live for 4,000 or 5,000 years. That's one generation time. So lodgepole pine has been in the Uintas only the lifespan of 1.5 bristlecone pine trees. So it's pretty neat when you find out, hey, this is a really dynamic forest system. It hasn't always been this way. And there's a shuffling of species. And these species, just like you and I, we like the thermometer in our house set at 70, right? We don't want it too hot. We don't want it too cold. We want it just right. It's the same for every organism. They want it just right. They want their abiotic, their non-biological conditions met. And then they have to deal with their biological challenges of predators and competitors and symbiotic relationships. And so they have to meet their conditions and find just the right condition to grow on. So the Uintas have been a great place for me to kind of study how trees and other plant species change through time, when they arrive. Why is it? Like what's the reason for them to arrive? So that's when we start to do, sometimes we call it single factor analysis. So you try to look at one thing at a time that could be a limiting factor. So we might look at, winter temperature. Is winter temperature, is it too cold in the winter? Or we might look at summer temperature. Is it too warm in the summer? So we start to try to build a story through single limiting factors. And that's often in research, we publish if we learn something about a factor. But the interesting thing is for almost all organisms, when you think about where they live, their range, so their location on a map, the kind of leading edge, so we could say the north edge of their range could be a abiotic limiting factor, could be temperature. The southern edge of their range could be a biotic factor, could be predation or competition. So even though a lot of the research I like to do is trying to narrow down to singular factors, we know that there's multiple layers of factors, and that you really have to try to find the evidence for each one of those factors. And in my work in fossil plant research, as you go back in time, 6,000 years ago, we don't have weather stations. So I can't tell you what the winter temperature was 6,000 years ago unless I use a model that somebody has developed to model back in time based on what we know about the temperatures plants grow in today. So, there's kind of a roundabout way to add more evidence as you go back in time when you don't have that direct observation. And a lot of that is with modeling. So yeah, there's, so the work I do is really, it's about, trying to uncover stories of the dynamics of ecosystems and how they respond to changes, what the mechanism is. And I don't think I said this, but lodgepole pine arrived 6,000 years ago. At the same time, wildfires began to really increase in the Uinta Mountains. So we think that's one of the mechanisms. Lodgepole pine has a cone that stays closed. It's a serotinous cone. It opens under the heat of a fire, so it's adapted to fire. And as fire increased, lodgepole pine arrives. So we have at least one reason. We don't have all of the explanations, but so really, the task of any scientist is, find the pieces of evidence and build an argument that supports the evidence, and then throw it out there to the community and say, hey, can anybody else reproduce this? Can you guys find a similar story if you did this yourselves in another location or even the same location? So it's it's evidence and it's replication. And I get really concerned when we find amazing results and then we can't replicate the results. That's a challenge because you don't know how strong that evidence is. So
I know I feel like everything you shared is just so incredibly helpful, and often students are, we know they're presented with a sound bite of science that's just like there is a fact, and it's so helpful to just hear all of this sort of big picture that you are sharing and the reasoning and community input. We are almost out of time. I have one more, hopefully quick question. It was so interesting to hear about the data analysis sort of portion you were just talking about. How do you actually go about collecting these samples? Like what does that actually look like? Maybe just paint a picture of how you go about doing that in the field.
Yeah, so what we typically like to do, so for like fossil plant preservation, the challenge in preservation in nature is that oxygen, air, often destroys plant material. It oxidizes and decomposes. So we try to find places where there's very little oxygen. And the best place to go is to the bottom of a lake, because lakes are what we call anoxic often. Those lower, lake levels, the mushy mud that squeezes out through your toes when you walk in a lake. Like, to me, that's where all the fossils are. That's the good stuff. And so what we find when we go out there is we have to figure out how deep is the lake. How do we get out to the middle of the lake? So we often want to go to the center of the lake. We want to collect a sample from the deepest part of the lake. So then how do we reach the bottom of the lake? So we connect, rods, and we have, I like the example where you have a milkshake and you put your straw in the milkshake and you put your thumb on the straw and you pull it out and your milkshake sample stays in the straw. So we have a coring device that's like a stainless steel tube that effectively has a mechanism like your thumb on a straw. So we push it in, we put the cap on it, and we pull it out, and it gives us that sample. And that sample is rich with fossils. We find twigs and needles and seeds and pollen and occasionally some bug parts. So there's so much to learn, when we start to look at these on the microscope. So yeah, it's really like getting out on these lakes. It's often, I'm with grad students and undergraduates and volunteers, and they do most of the heavy lifting, and I take notes. So that's kind of how we go about it. And then most of the serious research happens back in the lab. As we get things on the microscope and start to explore the world of fossil plants.
Excellent. Well, thank you so much. And that pretty much wraps up our Q&A for today. Of course, I want to thank Mitch and also all of our friends and scientists and everyone who has joined us along the way while we've been doing Research Quest Live. We continue to value feedback from our audience, and we're going to continue to make improvements and be thinking about this program in future iterations. So today really wraps up 3 months of Research Quest Live classes and Q&As. And if you're interested in continuing the learning or want to catch up with anything you've missed, of course, you can find all of those resources on the museum's website, the Natural History Museum of Utah, as well as on our Research Quest pages. You can also check out our summer offerings. We are rolling out a bunch of new stuff this summer. It's gonna be really fun. It's called Summer at Home, powered by NHMU. And thanks again, Mitch. And
Thank you. Yeah, please tell everyone to come visit when we reopen. I'd love to meet some of our viewers in person and have conversations. And it's much more dynamic to have a conversation with someone than to talk to a screen. So let's get those folks to come visit us when we open up again.
So true. Excellent. All right. Well, everyone, have a wonderful weekend and enjoy whatever weather you are experiencing right now, right? All right. Bye.
Bye-bye.